what are the reactants in PCR? So what do we need? What are the ingredients that we need? We need a DNA sample that we are going, oops, sorry, let me just do the pen. We need an actual sample of DNA, and we cannot amplify a DNA of any size that we want with PCR. It has its limitations as a technique. Make a note of that as a potential limitation for PCR, right? And we also require DNA nucleotides, a mixture of ATC and G. We also require the enzyme DNA polymerase. Note that DNA polymerase that we use in PCR is a polymerase that we isolate from a particular virus that is called TAC, and it has the unique property rather, that it is thermostable, and we're going to see why this is important. It doesn't degrade at high temperatures like other enzymes would. Finally, we also have something called a primer, which are short sequences of DNA. They're usually around 15 base pairs long. We can synthesize them artificially in the lab, and they are of known sequence. And they basically, we basically designed them to be complementary to the 3' and 5' end of the DNA sample that we want to amplify. So for instance, let's say that this is a chromosome. And let's say that I have a particular section of a gene that I want to amplify, which goes from here to here, for example, and it's around 1,000 base pairs long. I can design a primer that is complementary to this sequence and complementary to this sequence, such that when they extend, they can amplify for me the sequence in between. So the primers are going to sort of determine the sequence. They're going to flank the sequence that I'm going to amplify. Finally, I need the actual machine called the thermocycler, which will change the temperatures, literally cycle between different temperatures. Let's see what the stages of PCR are. So this here is going to be my sample DNA, or my template is another word. Sample or template DNA to be amplified. And the first step is called denaturation. I'm going to try and do it color-coded here for you guys. Denaturation is the first step here. So I go from double-stranded to single-stranded DNA. In order for denaturation to happen, I have to, of course, increase the temperature. We're going to see that exactly in a minute. The second step is annealing, where the binding of primers happens to the separated brand. Here is my primer in yellow, and it has bonded to the exposed single strand. And the same thing is going to happen to the other strand, right? It's just not shown here for simplicity of the diagram. And finally, I'm going to have the third stage, which is the DNA synthesis, or sometimes it's called elongation, where now the DNA polymerase comes along and actually, starting from the primer, will add nucleotides here, and it's going to synthesize the new strand, much like it does in DNA replication. What are the three main stages in PCR? And a very sort of simple one-marker question. So let's again do the usual thing of breaking down our question into context, command, and direction. Our context here is PCR reaction. That's a part of the spec that we need to evoke. My command word is what are, which basically translates to states in a way, right? And what is my direction? Well, my direction is that I'm only going to talk about the 3 main stages and nothing else. I'm not going to go into more detail. So take a couple of seconds to think about what the stages are. So my stages are denaturation, actually, yeah, there we go. Denaturation, annealing, and DNA synthesis. Elongation is also, depending on the exam board. Elongation is also acceptable. What are the mechanisms? What exactly happens in each stage of PCR? So this will be, so the steps in the way that we described them before are suitable for like a state or describe question, right? Where you're just simply stating what they are. This bit would be more suitable for an explain question where we explain what are the underlying biological molecular processes that are taking place in those stages. So, as we said, step number 1, we mix the ingredients together in a little test tube and we place them inside the thermocycler. The first temperature that this thermocycler is going to go for, is going to heat up to, is 95 degrees. So this 95 degrees here is called the denaturation. And what happens, why does DNA denature? Because the hydrogen bonds break. Remember that it's the hydrogen bonds that keep the 2 strands together. They form between the bases A and T and C and G. 95 degrees destabilizes them, it breaks them. We now have our single-stranded DNA, which we are going to move to the temperature anywhere between 55 and 68. Just note that like different exam boards have slightly different temperatures or ranges of temperatures that they quote. In reality, what this reflects is that depending on what kind of gene or gene portion you want to amplify, you always may have to sort of tweak and optimize the exact temperatures to get the best yield out of your PCR. So don't worry like an excessive amount about the temperatures. Just remember what kind of ranges you're at. So over 90 for sure for denaturation, high 50s, mid 60s sort of for annealing or primer binding or annealing. So here is my primer binding here. That temperature basically favors the formation of hydrogen bonds again. So the primer is allowed to anneal. And then thirdly, we have the temperature which favors the thermostable Taq DNA polymerase to then elongate my DNA here and allowing the DNA strand synthesis to occur. So basically what PCR results in, the final product is that we have duplication of the same sample of DNA. Now, all of this, let me just change color. All of this is a single, we call this a single cycle of PCR. Typically, we run anything between 30 to 40 cycles in a typical PCR. So I'm going to get multiple copies of my initial DNA sample. We call this an exponential amplification of DNA. Why is that? Because in each cycle, the number of double-stranded of products, of PCR products, double-stranded DNA molecules, doubles, right? How will that look like? So basically, one molecule will give rise to 2. 2 molecules will give rise to 4. 4 molecules will give rise to 16, and so on. So we call this an exponential amplification. And how can we calculate the number of expected products of double-stranded DNA molecules? We use this simple formula. Oops, sorry. we use the simple formula where n is 2 to the xth power, where x is the number of cycles that I run, and n is the number of DNA molecules that I end up with. Each cycle takes around 2 minutes only. So I can run, usually people run a PCR reaction for, a couple of hours, so several cycles. So you literally get millions or even billions of DNA molecules produced in a few hours. Let's see an example question of that. A PCR reaction is used to amplify a sample of DNA. The reaction begins with 16 molecules of DNA, which is a really low amount, and 23 cycles are completed. Calculate the final number of DNA molecules produced after the PCR has finished and give your answer to the nearest million. Okay, let's do our usual technique of breaking down context. Our context is PCR. Calculations. Our command words are to calculate and to show our working. Calculate and show your working. And the direction is that we are going to use this formula, even though this formula is not explicitly, we're not explicitly told it, but we have to know that formula. So take a minute to see if you can calculate that before I show you the answer. So let's do the calculation together. What is the formula that we're going to use? So as we said, the n, which is the number of product. Don't confuse this with the number of starting DNA molecules. This is going to be the number of product that we're going to get at the end. Is equal to the xth. Now let's substitute. So it's a good idea to always, show the examiner that you're applying the correct formula and how you're applying it. So we have a starting amount of 16 molecules of DNA. So 16, so we do 16 molecules of DNA starting. So therefore, 16 times 2 to the xth is going to be the final number of products that we're going to get. We are running 23 cycles. So we are going to have n is going to be 16 times 2 to the 23rd. Why do we multiply 16 by 2 to the xth? So this is our simple formula, right? So this simple formula tells us what? That if I were starting with one DNA molecule, then my number of product would be 2 to the power of x. Since I'm starting with 16 molecules, though, I have to multiply that by 16. If I go ahead and make that actual calculation, the answer that I get is 1, 3, 4, 2, 1. If you put this in the calculator, that is 7, 7, 2, 8, right? And I need to, so dot, dot. So I need to quote this to the nearest million. So if I round this up to the nearest million, here's my million. So it's basically 134 million. Okay, so they're testing like my numerical values as well, my numerical reasoning. What are the applications of PCR? So whenever you're also kind of like, outlining or describing an experimental approach to something, you will often see that the first mark in those questions is that you start with a PCR, because you will usually, if you extract DNA from a cell, you can only extract quite low amounts. So whatever you do anything, you will first amplify your DNA simply so that you have enough material to work with. So what can be used for PCR amplified DNA? Well, we can use that DNA to sequence it. For example, in determining mutations that may cause cancer in a patient. I extract DNA from my patient, but I'm going to get a very small amount of DNA. I'm not going to keep calling the patient back for extracting DNA. So I'm going to PCR amplify it first. We can use DNA in profiling or fingerprinting to, for example, match DNA from crime scenes or for paternity testing. Or we can use it for genetic engineering, such as for making genetically modified organisms, whether they are crops or whether they are, for example, bacteria expressing the human insulin gene. So let's see an example question. Explain the importance of primers in the polymerase chain reaction for 2 marks. So my context again, of course, is PCR. My command is that it is an explained reaction, but it's a short explanation, right? Because it's only 2 marks worth. And my direction is that I am stating the importance of primers in PCR. Take a quick minute to see if you can figure out what each mark is for. So primers are complementary to the 3' end of each separated strand of DNA, allowing them to anneal. So that's one thing that they do. It's 1 mark. This creates a double-stranded area which Taq DNA polymerase can bind to, allowing DNA synthesis and elongation to occur. DNA polymerase cannot start adding nucleotides to each other, polymerizing nucleotides from scratch. It usually needs to have a short little sequence of double-stranded DNA to be able to add nucleotides to. So this is what we mean here by create a little double-stranded area which Taq polymerase can bind to, allowing DNA synthesis to occur. Also, it prevents DNA strands from rebinding to each other. So once I have done my denaturation steps and the 2 strands have separated, when I lower the temperature back down for annealing, theoretically those 2 strands could just anneal back together. And a little bit of that will happen, right? Which is why the calculations that we do previously are the theoretical maximum yield of a PCR. In reality, we might get slightly lower product.